students, today we are discuss about shell, the unit of life. Shell is the unit of life. What is shell? Shell is the structural and functional unit of life. What is the structural? It means each and every shell a fixed shape, each and every shell a fixed function. So cell is the structural and functional unit of life. Every shell has its own life. Each and every shell has its own life. All organisms, including ourselves, start life as a single shell called zygote. It means each and every organism, including me also, ourselves also, start life as a single shell, and this single cell is called the zygote. Zygote is the diploid. Zygote is diploid. It means after the fertilization. And one thing is the haploid. N number of the gamete. Haploid. N number of the gamete. Haploid. 2n number of the gamete. Diploid. Next point. The invention of the microscope and the discovery of the shell. At the first time, invention of the microscope. The first microscope was constructed by a Dutch scientist, Antony van Leeuwenhoek. So Antony van Leeuwenhoek constructed first microscope, and this scientist is Dutch scientist. This microscope consisted of a single biconvex lens, and we are called simple microscope. Robert Hooke, an English scientist, developed a microscope by using two lenses for achieving greater magnification. This is the greater magnification. Such a microscope known as compound microscope. Robert Hooke, 1635 to 1703. Robert was an English scientist, and this scientist developed a microscope by using two lenses for achieving greater magnification. So that microscope is called compound microscope. The invention of the electron microscope magnification to over 2 lakh times against the ordinary compound microscope. It means the electron microscope magnification more than 2 lakh as a comparison of the ordinary compound microscope. So, which magnify an object up to maximum about 2000 times. 2000 times maximum. Then next, the ordinary compound microscope uses light, which is bent by glass lenses to magnify the image, and the electron microscope uses beams of electrons. Electron microscope uses beams of electron which are bent by magnets. So the ordinary compound and electron microscope. Ordinary compound microscope uses light which is bent by glass lenses to magnify the image. But the electron microscope uses beams of light which are bent by magnets. Shell theory. What is shell theory? First, shell theory proposed by three main scientists, M. Sliden, Schwann and Virchow. The main concept of the shell theory, number one, the shell is the smallest unit of a structure of all living things. Cell is the smallest unit of a structure of all living things. Second point, the cell is the unit of function of all living things. First point, shell is the smallest unit of a structure of all living things. Second point, cell is the unit of function of all living things. And third point, all cells arise from pre-existing cells. All cells arise from pre-existing cells. And these three different points proposed by three different scientists. So in 1838, Matthias Schleiden. This Matthias Schleiden is a German botanist. And this German botanist saying that every plant is made up of a large number of cells. Matthias Schleiden, a German botanist, and this German botanist saying that every plant is made up of large number of cells. After 1 year, Theodor Schwann, a German geologist, made similar discovery in animals. It means each and every animal made up of large number of cells. In 1858, Rudolf Virchow saying that all cells arise from pre-existing cell. So three different scientists, Matthias Schleiden, Theodor Schwann and Virchow, proposed a theory, and this theory is called shell theory. According to shell theory, these 3 points. First point, the cell is the smallest unit of a structure of all living things. Second point, the cell is the unit of function of all living things. And third point, all cells arise from pre-existing shell. So it is also called the shell theory. Shells, how numerous? Single shell. Many small plants and animals are made up of single shell. For example, bacteria, yeast, amoeba, all are the single celled organisms. Fusion. Some small plants and animals are made up of relatively few cells, just a few hundred or few thousand cells. For example, Spirogyra and Volvox, etc. Number 3, multi-cell or multicellular. Most plants and animals are made up of millions and billions of cells. Millions and billions of cells. For example, human beings or Homo sapiens, mango plant, etc. Next, cells, how small? Number 1, smallest cell. Smallest cells are the bacteria, measuring about 0.3 to 5.0 mu. Mu is equal to micrometer. RBC about 7 mu in human body. 7 mu means 7 micrometer. Longest cell. Cells are nerve cells. Nerve cell is the longest cells in our body. Largest cells are bird's egg. Actually, the central yellow sphere. In egg, a central yellow sphere is called the egg part. For example, ostrich egg is the largest cells in the world. The white albumin of the egg and the egg shell are extra parts. So this is the albumin and the eggshell is the extra part. Next, cell cycle. Human RBC are circular and biconcave. To pass through narrow capillaries and transport oxygen. So human RBC are circular and biconcave to pass through narrow capillaries and transport oxygen. Number 2, WBC, white blood corpuscles. WBC, white blood corpuscles. RBC, red blood corpuscles. WBC are amoeboid. It means amoeba-like structure that can squeeze out through capillaries wall. Nerve cells are long to conduct impulse up to the brain and vice versa. Muscle cells are long and contractile to pull or squeeze the parts. Guard cells of stomatal pore in the lips are bean shaped to open and close the pore. So guard cells protect the stomatal pore, and this stomatal pore opening and closing by the action of guard shell. So today we are discussing about only the introduction of the shell, cell shape, invention of microscope, etc. So the next video is structure of cell and its function. Okay, thank you.